Hi, my name is Faith, and I'm a computational cancer genomics first-year PhD student, and I usually work on breast cancer, but for this hackathon, I really wanted to challenge myself with a rare underserved one. In the spirit of Anthropic's focus on neglected diseases, I chose diffuse midline glioma. It's uniformly fatal and has a less than 1% 5-year survival rate, so the question of what to test next still matters enormously. In DMG, the drivers hide in the non-coding genome, the switches, not the genes. And today you get a score from one tool, a motif guess from another, an evolutionary constraint from a third. None of them are in your tumour cell type, and none of them are coupled. And validating even a single non-coding variant at the bench is slow and expensive. So the real bottleneck was never prediction. It's deciding which handful are worth it. NCypher does 3 things in one agent call. It tells you which non-coding variant to validate first, it shows the exact base and motif that break, and it flags honestly when the models disagree. Coupled, so you get to the bench sooner. I started with 10,869 somatic non-coding variants across 152 diffuse midline glioma patients. Naive recurrence just finds artifacts, so I ranked by function and a matched background. The method couples 3 independent axes: chromatin accessibility from the developing brain, evolutionary constraint from phylopy and zoonomia, and measured function from developing cortex MPRA. The MPRA was actually where I started, and it gave an honest negative that redirected me to caQTLs, the right ground truth for Chrome BPNet. 164 converged, and one kept rising to the top. NPAS3, a neurodevelopmental tumour suppressor, where a switch inside the gene loses accessibility at the exact conserved base. Every step is kept honest in a versioned second brain, a context harness so the work survives long sessions. And when Alpha Genome disagreed on NPAS3, I didn't bury it. I downgraded my own lead and said so, and I looked at why. Alpha Genome predicts reference context tracks, not the patient's tumour cell state, which is likely why it disagreed. The disagreement can be a signal too. Here's the real thing running in Claude science. I hand it a structured brief, it plans the work, and it scores its own confidence before it runs. Subagents call the NCypher tool. One call returns the rank shortlist, the mechanism, the motif, the exact base, and the saliency picture, not 3 lines of text. Then, a second agent rederives every headline number from the raw files, an agent checking an agent. The heavy scoring and the fetal brain high C run on modal. And on every push, GitHub Actions has Claude rederive the headline number from raw data, so the finding stays reproducible. A finding is only as strong as the test that can break it, so I tried to break mine 6 times, each in its own Claude science session. Confounds, whether the signal is really brain-specific, whether the model learned real motifs and not a GC shortcut, an independent second opinion, whether the axes are truly independent, and real fetal brain high C. The same engine triages the whole cohort. In a rare disease where samples are scarce, every extra validated lead counts.